Okay, so some concluding remarks that I have about PCA. So the things that we covered here are that we have this dimensionality reduction technique that uses lines, so it's a linear dimensionality reduction. Can be done through a technique called PCA, which we've covered here. And so PCA can typically be done through a mathematical operation called the singular value decomposition, or SVD. And we've also looked at some uses of PCA in genomics. And so some things to consider from this is that PCA is not without limitations. So earlier we talked about how PCA requires linear relationships, so that's one example. So if your data is not linearly characterized well, you may want to use other types of dimensionality reduction techniques. And the other thing is that PCA also has many variants or flavors. So two examples of that are commonly used are the sparse PCA and non-negative matrix factorization, or NMF. So if you're considering using PCA, you may want to think about some of these limitations, or if there's a special version of PCA that better suits your needs. Okay, so just to end, I'd like to again thank the UCLA Computational Medicine Department for supporting and funding BigBio. I have answers to the worksheet after this.
That's good. I have a few questions first.
Okay, sure.
What is a singular value?
Okay, so the question is, what is a singular value? So in this kind of case, we don't have to really consider what a singular value necessarily is in a mathematical sense. But in this case, we're using it as a way to explain the amount of variance. So in this case, or in the context of PCA, we think of a singular value as a number that kind of measures the amount of information or variance that a PC carries.
And then similarly, how do you tell if your data is well characterized by the linear?
So like one way that you can do that is you can look at okay, so the question is, how can you tell whether your data is characterized well by lines or linear relationships, so whether PCA is a good fit for your data? So one way is to simply run PCA and see if something good comes out, or if you don't want to actually run it, you can look at like subsets or plots of your data and see if it follows a somewhat linear relationship. But there's not really...
Something good look like if you ran it? Sorry.
So ideally what you'd want is that your PCA clearly separates out some sort of groups of interest for you. But it's not really easy to say whether PCA will work for you or not without actually like looking at it, unless there's like some sort of domain information known for your specific study that PCA is known not to work well. Okay.
Then let's do the questions.
Okay. Okay, so I'm probably just going to go through it because a lot of it's like plots and stuff like that.
Yeah, well, and also I don't think that we'll be able to hear people. I don't think that worked yesterday. So
okay.
You should just do it.
Okay. Okay, so now we're going to go over some of these worksheet questions. So the first question is, what are some of the uses of dimensionality reduction? So we went over a lot of different uses. And so some more general ones are like visualization or pattern discovery, could be preprocessing, noise removal, compression, or decreasing computation. Or if we're specifically looking at some uses in genomics, we again kind of mirror what those general uses are, but you can be looking for things like ancestry, match effects, geography, or you may use it for some preprocessing of some other analysis. Okay, so the second question is, you're given this plot and you want to draw the first and second principal components. So one way to approach this is to look at this plot and see what kind of direction has the most variability, or what's the largest spread. And then with that, you might guess that the first PC is kind of diagonal across the flat region of this blob. And because we know a few things about principal components, so one way to discover the second one is you might think that it's independent. And so if we remember from basic geometry, right angles are when things are perpendicular or orthogonal. And so that's why it forms this cross formation. The other way to think about it is if we've captured all the information along this axis, where would the rest of the information be? And that can be found by creating something vertical to that. Okay, so the third question is, given the following singular value matrix, what percent variance does the second PC explain? And it's okay to leave this as a fraction. So we're given this formula here, or this matrix here. And if we recall, there's this formula that tells us how to calculate that. And so if we're interested in the second PC, we'll take 2, we'll square it to get 4. And then to get the denominator, we'll take each of these entries, square each of them. So 3 squared plus 2 squared plus 1 squared. And we'll take the fraction of that. So ultimately, after you do some arithmetic, you'll end up with 2/7. OK, so the next question is, what would the value of the second singular value be in the following scenario? So this is actually the same example from these slides here. And you notice that there's these lines arranged in a diagonal line. And so what might happen first is that we'll fit this diagonal line. And now the question is, well, we've captured everything because it's on this diagonal line. So there's actually a trick to this, and it's actually that the second PC actually has a singular value of 0 because we've explained all the variation in this data. And so therefore, the second PC gives us no new variation, and that's why it has a singular value of 0. OK, so the last question is, what is a pattern that can be pulled out by PCA and genomic data? And some answers for that are ancestry, batch effects, and geography. And there are many others, but these are some that we gave as examples in the presentation. OK, I think that's it.